It's an enormous honour to obviously be awarded the Phelps Foundation Medal, and I hope to do it justice in terms of delivering on the future promise in my career. So I wanted to tell you today about some of the work that we've been doing, not actually to do with sleep or Parkinson's disease or anything like that. It's actually very, very basic research that we hope in the future will translate into medical care, and I'll come back to that right at the end of my talk. So it's probably not escaped your notice that we live on a planet which has an intrinsic period because of the rotation of the Earth. And that rotation has basically programmed everything on the planet to have a kind of reciprocal rhythm embedded within it. And we know that organisms all the way from bacteria to complex organisms like us have these circadian clocks. And in us, there are a variety of different rhythms that you'll recognize or maybe even take for granted that occur on a 24-hour basis. So every day, even before you wake up, your body is basically getting you ready for the day by upregulating your hormones, such as cortisol, your blood pressure, getting you ready for the day ahead. And then at night time, it's putting you to bed in the same manner by changing your temperature, secreting various hormones to coordinate this. And this is done proactively and not reactively. So the reasons we have a biological clock is that we can preempt what's coming, because after all, the daily cycle is eminently predictable and occurs every single day. So why not have a system that actually is able to predict what's coming if it's going to come every day? And that's in fact why the clock exists, to anticipate stuff that's going to happen before it does happen, so that we're actually able to better utilize our environment. And that's proven by an experiment I'll come to in a second. But before I come to that, I'll just tell you a little bit about how clocks are distributed through the body. If I was talking to you 20 years ago, I would have very much been talking to you from a neuroscience perspective, because we believe that the clock basically resided in the brain in a tiny little nucleus called the suprachiasmatic nucleus, consisting of about 50,000 neurons in us. But it turns out actually that that's not the case, and in fact every tissue within the body, and in fact every single cell, even skin cells like fibroblasts, have the capacity to exhibit these circadian oscillations, these daily oscillations of about 24 hours. So how do we know that they're important? Well, this is an example from a mammalian system, but people have done similar experiments in plants and bacteria and even in fruit flies. And this is from an experiment done in chipmunks. And what they did in this experiment, which was done in the early 2000s, actually the real experiment was actually done in the 80s, this is from a review. The suprachiasmatic nucleus, which I told you about just now, if you knock it out by doing an electrolytic lesion, you can basically convert an animal with a very robust rhythm. Here the black stuff indicates activity, the white stuff indicates rest. You can basically turn that very rhythmic animal into an arrhythmic animal. So its overall activity is not that much changed, it's a bit lower, but it's basically distributed all throughout the day, so it has no idea what's going on. Now, if you put animals with lesions back into the environment with sham controls that have had surgery but not actually knocked out their SCN, you can see that they are outcompeted very quickly and they die off in the environment. And as I say, people have done this with various different organisms and shown the same thing. So it has a survival advantage, and that's probably why we've kept circadian rhythms going in complex organisms, even like us. The thing is, it's very difficult to see the effects of these things because we, as humans, have become so good at controlling our environment that we no longer are hostage to the environment like our kind of cousins were in the distant past. So clocks are also now obviously implicated in many diseases because we have these clocks absolutely everywhere in our bodies, not least in our brains where they're associated with neurodegenerative conditions, in the heart, in the immune system, and particularly in the metabolic tissues such as the liver, pancreas, muscle, and fat. So clocks exist everywhere, and normally they're coordinated in order to make us better physiological machines. And if that goes awry because people do shift work or get jet lagged, then you can upset the normal patterning. And we know that that's really important for predisposing people to metabolic disorders now, and also in particular to cancer. So there's very good evidence for that. The other stuff, there's less good evidence for at the moment, but it's emerging with time. So what my lab is particularly interested in is how these clocks actually work. Now, the fact that they work at a single cell level is actually pretty useful for us because it means we can really confine the problem to looking at individual cells. So this shows some cells in a dish, completely separate from the world. They're in a dark incubator at a constant temperature, so nothing is telling them what time of day it is. But these guys have been transfected with a reporter that basically glows during the daytime and is dim during the nighttime to them, so their subjective time. And they're able to tick and tock like that without any intervention for days on end. And in fact, tissues can be looked at in the same way, and they can stay like this for up to a year in continuous culture, ticking and tocking away. So how does this work? So the model that's been proposed over the last 20 years, built on observations in Drosophila, is basically, it revolves around something called a negative feedback oscillator, which is probably how all oscillators work, whether physical or biological. And in this model, you have a positive regulator, which switches on clock genes. These clock genes then get made into their mRNAs and eventually into their proteins. And then the proteins then negatively feed back to switch off the positive drive. And then gradually, as the clock genes aren't stimulated to be reproduced anymore, they switch off. The negative regulation eventually goes, and then the whole process resets itself and the clock genes turn back on and another cycle starts. So this whole thing takes about 24 hours to work, which defines the circadian clockwork. Now in mammals, the players in this circuit are shown here. So the positive drivers are these transcription factors called clock and BMAL, and the negative elements are these genes or proteins called period and cryptochrome. Now, a similar thing happens when you look at the fruit fly, which is where most of the model was developed, and you have similar positive drivers and negative suppressors on the other side. But you'll notice there are differences creeping in as we step back in evolution. Now, if you look at plants, it's a completely different ballgame. So all of these components now are completely different. There is no homology between these components and the ones in fruit fly or us at the genetic level or the protein sequence level. And if you go back to the furthest cousin of ours, if you can call them a cousin, the cyanobacteria, again, there is no homology between these proteins and genes and plants or us. So there doesn't seem to be any overlap in these basic pathways. So if you kind of summarize that, there's a lack of conservation, evolutionary conservation, in the transcriptional oscillators that we know exist in these model organisms. And these are basically the 5 principal model organisms that we study clocks in. Different researchers around the world study them. But you'll see that the clock genes that we've kind of been looking at over the past few years aren't the same. So this begs the question as to, is this transcriptional drive actually important for sustaining circadian rhythms? There are lots of other observations that I haven't got time to go into why this transcriptional oscillation might not be the whole truth, but an experiment that we did to try and look into this in further detail was to actually use red blood cells. And we thought, what's the best way to actually look for non-transcriptional oscillations? We'll pick the right model and then try and show or try and prove or disprove what we think might be the case. And red blood cells obviously are known for their ability to transport oxygen around the body and are packed full of hemoglobin for that purpose. But also they're actually a great model because they're basically a bag of cytoplasm, and they don't have any organelles and they don't have a nucleus and they don't have the ability therefore to perform transcription. So we can use this as a model to inform us about whether non-transcriptional oscillations can occur. The problem is, as soon as you try to ask this question, you run into a problem, because all those proteins that I've been telling you about, clock, BMAL, period proteins, they just don't exist within the red cell. So even if we wanted to simply just look at the same stuff that participates in this transcriptional loop, we can't do it because the stuff isn't there in red cells. So we ran into a problem. But thankfully, we also had a solution to this problem by looking back at some of our previous work. And that work was done in 2006 while I was a postdoc in Mick Hastings' lab. And back then we were trying to look at proteins that oscillated within the liver. And it turned out in this study we found that 10% of all proteins in the liver oscillate, and that's actually known now for multiple tissues in the body. So every day 10% of your proteins in any individual tissue will undergo a biological oscillation. And we found that obviously interesting from a global point of view, but what was really interesting was one of the proteins that we found during this experiment looked even more interesting because it exhibited a post-transcriptional, or in this case, a translational modification that occurred on a circadian basis over 24 hours. And this is a gel, don't need to worry about the details, but it basically has 2 spots on it. And basically those 2 spots are different because one of them, the one in blue, is basically oxidized, and the one in red is not oxidized. So they're basically the same protein, but they're moving between 2 different forms. And you can see that on the graph here. The blue one does that, and the red one does that. They're in antiphasic oscillation. What's happening is they're basically shuttling between the 2, but the actual level of the protein isn't actually changing. And that's important because if that was the case, we could also potentially use that same marker in a red cell which can't make new protein and look at oscillations. And it turns out that that's a well-trodden path in a different field, and antibodies exist to actually probe for this oxidized form of protein. So just a little background on these proteins, these peroxiredoxin proteins. They have cysteine residues which are oxidized by hydrogen peroxide, and then they can be further oxidized into what's called a hyperoxidized form, and that can be recycled back to the initial state by an enzyme called sulfurredoxin. And we, as I said, could probe this bit using an antibody and probe this bit using a different antibody, and therefore work out cycles in red blood cells. So we did a simple experiment. We took blood from people, purified it, and then basically incubated it in the dark at the same temperature, so it had no idea what was going on in the external world, and then mapped their oscillations over time. And when we did that, we found a triplet of oscillations within the red blood cell. We observed oscillations in the peroxiredoxins. That oxidation reduction reaction that I was telling you about occurred within these red blood cells, which obviously was incredibly surprising. We didn't expect to find that. And what was even more impressive is that when we thought about where this hydrogen peroxide might be coming from within the red cell, we figured it was probably emerging from oxidation of hemoglobin that occurs within the red cell. So one of the reasons why we were really interested in peroxiredoxins is that because they're highly expressed in red blood cells, and they exist there to mop up hydrogen peroxide that's actually being created by this process called auto-oxidation, which happens in red blood cells. And one of the things that you see in peroxiredoxin mouse knockouts is that the red cells become unstable and that you actually get hemolytic anemia. The red cells actually break down. And they're actually there to protect the red cell from this oxidation process, this hydrogen peroxide flying around. So we figured, is there something going on with the hemoglobin? And we looked with a different assay based on fluorescence, and we found that, again, you could see these oscillations in these isolated cells with no idea of what's going on in the outside world. And we figured that reduction back to these forms that could be used to carry oxygen would also be going on, and that's dependent on reduction factors within the cell, these archetypal reduction factors, NADH and NADPH, and indeed they exhibit circadian oscillations. So by 3 different methods, we were able to show that in these red cells you had pretty robust oscillations that couldn't be explained by the current model of how we think the clock works. Now, peroxiredoxins aren't just in us, they're in practically everything on the planet. They're incredibly conserved proteins, all the way from bacteria, even in archaea, the third kingdom of biology, and also in eukaryotes like us. And what we were really lucky with is that the antibody that we used to probe those oxidation reduction reactions was actually, because the peroxiredoxin is so highly conserved around the business end, the active site that actually takes part in the hydrogen peroxide metabolism, we're able to use that same antibody potentially in multiple different species. And that's what we did, clubbing together with basically all of our friends who we could get organisms off around the world. We basically mapped these oscillations in a variety of different organisms, including prokaryotes and eukaryotes, so bacteria, even archaea we managed to get hold of, flies, fungus, plants, whatever we got hold of, we found that we could see these circadian oscillations in peroxiredoxin oxidation reduction rhythms, and even an algae that we managed to get hold of through Andrew Miller in Edinburgh. So in virtually all species that we were able to, all the species that we were able to get hold of, at least in terms of model organisms, we were able to show robust circadian oscillations of these peroxiredoxin proteins. So although you don't have conservation of the transcriptional elements, the clock genes, we showed that you could have conservation of these peroxiredoxin rhythms, which really makes you think that the peroxiredoxin side of things, or at least redox reactions, are more fundamental in terms of the oscillations that are occurring than the clock genes that we previously focused on. And that's really important because it means that we might be able to get further insights into how these mechanisms actually work, which is what my lab's basically focused on at the moment. So we have to kind of rethink how the clock looks. So everything I've told you in the first part of the talk was about this transcriptional translational feedback loop. But in fact, that's important. We know that through some of our experiments as well, so that we can't just chuck this bit in the bin, but also we have to factor in this bit, this new bit that we never even knew about before. And that's because in the complete absence of the nucleus in the red cell, we could still see these robust oscillations of reduction and oxidation within those cells. So why is that important? Well, transcription factors are actually pretty tricky to target in terms of drugs, for example. So targeting those transcription factors such as clock and BMAL is pretty tricky, and they look very similar to other transcription factors in that same family. There's about 160 within that superfamily of transcription factors. So specifically targeting the clock ones is actually pretty difficult. But peroxiredoxins are enzymes which are potentially more druggable. So this offers us a kind of way into the clock. So one of the things that we've been trying to do recently is to actually target peroxiredoxin molecules using a virtual drug screening approach. This is a structural representation of peroxiredoxin 4, one of the homologs, and painted red is the active site region. This is actually 5 of them joined together, then actually form a donut in a kind of polymer of 10 normally, and this is one of the stable forms that you can actually crystallize. So what we did was to use our supercomputing cluster in Cambridge to actually dock drug molecules into that red pocket virtually. So we used about 8 million compounds and docked them in virtually to see which ones would fit best. And these are the top 100 that you can see docking into the active site. And it took about 200 computers 3 days to do what you're seeing here. With 8 million molecules, this is showing the top 100. But these are the best fit ones. So we've isolated about 50 of those and started doing high throughput screening, or relatively high throughput screening for us, to try and see if these actually have any biological effect. Because it's all well and good having virtual screening, but you want to actually prove biological function. And we've done that with some assays that are quite commonplace in the circadian arena. We use these 384 well plates. Each one of these wells is basically a collection of 5,000 cells that we're looking at from above, and each one of them is expressing one of these circadian reporters that I showed you oscillating within those individual cells. And these are actually 2 plates with exactly the same conditions on them. So some of them have drugs in, some of them don't, and actually they're expressing reporters that are actually antiphasic to each other. So if you look carefully when I play the video, you can see they're actually kind of oscillating in antiphase. So what we can do is we can take pictures of this over time and then quantify those individual dots and see what happens when we throw on these drugs. And that's our kind of assay for whether they can have effects on the biological clockwork. And if you graph that out, this is the raw data on the left. This is what we call the detrended data, so we can see patterns more easily. Most of the drugs do this. They don't do anything over wide concentration ranges. We usually do 10 concentration ranges, but they don't mostly do anything. But some of them do something. In this case, prolong the period. You can see the black line here is the control, and this one here is treated with a drug. And likewise, in a kind of dose-dependent manner, down here, a completely different drug, which is completely novel, can affect biological clockwork. So we're hoping that by using this type of approach, we can actually develop molecules that can adjust the clock, either prolong it, dampen it, or shorten it to do what we want it to do. And as I say, this is far more promising than any previous approaches that we've had available to us, because you can actually target these molecules in a structural way, which is pretty important for finding possible therapies. So I just want to end my talk there and acknowledge the people who did the work. I did some of it, but not all of it. People in my lab: Rachel Edgar, Utam Velakunja, Alessandra Stangerlin, Ian Robinson, Guillaume Ray, Lisa Wu, Nick and William, who are PhD students and rotation students at the moment, respectively. And then a variety of different labs across the world and across the country that made this work possible. And obviously, immense amounts of funding from the Wellcome Trust and the ERC, who bankroll my lab at the moment. Thank you very much.